Hello, everyone joining us. Welcome to our second RDCA-DAP webinar of 2023. We're just going to allow another minute or so for everyone to join, and we'll get started in just a bit. Thanks for joining us. Good afternoon and welcome, everyone. My name is Dominique Cruz. I'm a project manager with Critical Path Institute, and I'm pleased to kick off today's webinar, GUIDs and De-Identification Tools for Rare Diseases. So I'm going to start off with just a few housekeeping items. If you have any questions throughout the presentation for our speakers or about any of the topics that are discussed in general, please put those in the Q&A box. The chat function is disabled for everyone, just so we can make sure we aren't missing any questions that pop up. So if you have any questions throughout the presentation, please put them in the Q&A box and we will address them. Also, just a note, all of the participant lines are muted to reduce background noise. We're also recording this presentation, and that link will be available via our YouTube channel and shared shortly after the presentation. I've also included our general contact email address. So if you have any questions or want further information once the webinar is complete, feel free to reach out to us there. At this point, I'm pleased to introduce our presenter. Saneth Kumar Ramesh is the CEO of the startup company CuresDev. In his career, he has built and launched several successful open source software products and is now using this skill set to enable treatments for 400 million patients worldwide affected with rare genetic diseases. I'm excited to hand it over to Mr. Ramesh, who will take it away from here.
Thank you. Thank you so much for having me. I'm excited to share my learnings over the last several years to this group. Everything started about 4 years ago after my charming little boy, Raghav, was diagnosed with a ultra-rare disease. He is one of 10 kids in the world with this condition. And when he got diagnosed, he was the only person that we knew of. It has been an amazing experience raising him as a child, but it has also been an incredibly painful and challenging experience at the same time. He is not able to sit, stand, walk, talk, eat anything by mouth. He has seizures. He has developmental delays. He has 11 different subspecialties that he's going to at our hospital here, but he's always smiling and happy. And that is what keeps us going. So after my son's diagnosis, I started a nonprofit focused on his treatments, raised about $500,000, and created 7 different treatments through drug repurposing for him. Many of them have had tremendous impact on his quality of life today. My background started in the tech industry. I am a software leader building healthcare and open source products. I started another nonprofit organization called the Open Treatments Foundation, specifically focused on using software to make a difference in the rare disease space. We are building an open source rare disease diagnosis counter, which was funded and supported by the Chan Zuckerberg Initiative. And I'm using that experience to break the data silos in rare diseases through my current company, CuresDev. Speaking of data silos, all of us have heard this term, and at every rare disease conference you go to, people are talking about data sharing. But I've asked a lot of patient foundations and biotech companies and academia on what it means when you say data sharing. And it comes back to something to the effect of, data silos are really undermining drug development. And so we need to share data among institutions for us to go faster, for us to get treatments sooner. By the end of this talk, you will understand what I specifically mean by data sharing, and you will have a couple of tools at your disposal to enable data sharing at a level that was previously not possible. But first, I want to highlight this paper that some of my friends and colleagues in this industry have written. When I read the paper during the middle of the pandemic, I thought, wow, this is bold. If you read the abstract, they don't go soft because of the magnitude and the impact of problems that data silos are creating in the industry. To illustrate the problem further, the paper has a quote from Tracy Dixon Salazar, who is a parent of Savannah that has a Lennox-Gastaut syndrome. I won't read the whole quote here, but I would highly recommend you all to read this paper once or twice, or even thrice, because there are a lot of deep insights that are being shared as a part that highlights the problem of data silos. To me, data silo is like this parable where several blind men are looking at an elephant or touching an elephant and trying to figure out what it is. Someone says it's a snake, someone says it's a wall, someone says it's a rope. But no one really has come up with the understanding that it's an elephant. For me, the entire rare disease knowledge base is like an elephant. And all of us are blind folks touching the elephant, different parts of it, and trying to make inferences. For example, a clinician who takes care of a rare disease patient might say, the quality of life of this patient is affected the most because of the developmental delays. This patient is not able to walk. They cannot go to school. They cannot read a book. And that is the problem that we need to solve. But if you ask the parent, they would say, seizures are what impacts this patient's quality of life. My son is not able to sleep peacefully at night because he's constantly getting seizures. He's not having a fun time with his friends because we are just afraid when he would just seize and fall. If you ask a researcher, they would say, this rare disease only has one pathogenic variant. But if you look at the natural history database, there would be 15 reported variants of either uncertain significance or likely pathogenic, which might just be a causative for the disease. This is just an example of how different parts of the rare disease elephant give you different inferences. And we have to put everything together, aggregate the data to actually have a holistic view of the rare disease landscape. And that is critical for us to unlock treatments for all of the rare disease patients suffering today. I got introduced into this problem when I asked this one simple question: how many patients does a disease have? I started asking this question around because I was very, very eager to find more patients for my son's condition. After his diagnosis, in about 2 months, we discovered 2 other patients. And we thought, okay, this is just the beginning. In about a year, we're going to have 500 patients worldwide. We are going to be breaking the barrier for gene therapy treatments. There's going to be a biotech that's going to invest, hundreds of millions of dollars. And my son is going to walk, talk, go to college, and live happily ever after. 4 years later, we only know about 10 patients. And I am personally only connected to 3 other patients. And so I started asking around this question for each of the rare disease communities: how many patients does your community have? And they'd say, my Facebook group says there's about 150 participants, so there's probably around 100 patients. They would look at their registry and say, no, there's about 250 patients. Someone would say that their clinician told us that they already have about 500 patients. There was just no clear answer. And this frustrated me because you could actually ask the people that are assigning diagnoses to these patients and aggregate the information that you get from each of them and count the number of patients getting diagnosed per year with rare diseases. And this is what I set out to build with Open Treatments Foundation. So hopefully at some point we will have that data. But right now, the challenge seems to be not in the conceptual understanding of aggregating data from diagnostic companies, but in the actual act of moving the data together and the technology that is necessary to make that happen. This gets to the heart of the question, what is data sharing? I want to be able to define things so I can deeply understand them. My definition of data sharing, which is very, very simple and rudimentary, is you are unifying data from one or more databases. The term database doesn't have to have any technical meaning here. It is just a collection of information about a patient, a disease, whatever. It could be an Excel sheet You could call that a database. It could be a Word document that you have a bunch of bullet points. That's the database. It doesn't matter. As long as you're unifying them into one place, I call that a data sharing. More specifically, I want to define data sharing with two components. One of them is linking. The other one is merging. And it took me a while to arrive at this conceptual understanding. And once I arrived at this understanding, it was a lot easier to have conversations with people. Linking is simply the ability to connect the data in the future. Let's say you have a diagnostic testing company that's offering a panel on diseases that have seizures. And you also have a database, a natural history database that's collecting data from patients that have seizures. Eventually, you would want to put these two together, right? To know if the patients that have reported information in your history actually have a diagnosis or not. Today, most databases don't have the ability to be connected in the future. You don't have to connect them together. There's probably no business need for it. There's probably no legal and compliance framework available today to do it. But eventually, when the time presents itself, you want to be able to connect this database together. And that is what I call linking, or the ability to connect the data in the future at some point. To do linking, merging is the idea of actually copying data together into a new location. This is what I would this is what most people refer to as data sharing when they think about data sharing, because you want to be able to share data among each other. But that's a harder problem to solve, because linking, all it requires is that you have a shared identifier between two databases. If Sanath has some information in database A and database B, you should be able to put information about Sanath together. And to do that, you need to have some shared information. And in this case, we call it a shared identifier. And you also need to have your IRBs, or your ethics boards, approve to having shared identifiers in place. This is a nuance, but I want to also highlight this nuance, because identifiers don't have meaning in and of itself. But when you have identifiers that sort of gets shared across databases, you run the risk of re-identification eventually when you put all of the data together. And so storing shared identifiers require extra IRB approval. But that's about it. What about merging? Merging actually requires patients to consent data, because you're physically copying data over, and someone is going to be having access to a larger corpus of data than they had before. It requires compliance with different privacy laws. It requires contract between institutions that are sharing data. I was once told by someone that it took 6 months to simply negotiate a contract so they can then talk about data sharing. It also requires, a good platform where you can share data among each other. There are a lot of considerations, including anonymizing data, de-identification of data, and making sure the combined data sets don't pose an extra risk to privacy. If there's one key takeaway from this talk, it is this: make your data sets linkable. Create enough information in your data set so you have the ability to connect them in the future. Whether or not the data actually gets merged together is not under your control today, or may not be. But as long as you lay the groundwork, you lay the seeds today, they will eventually grow into a big tree that bears fruits and hopefully makes the data mergeable. To make the data linkable, all you need is a shared identifier. And in the rare disease space, people call this a GUID. I'll talk in a minute about what GUID means and why that is not a good term. But all you really need to keep data linkable is a shared identifier. Now, what is a shared identifier, aka GUID? I'm going back to this quote from Tracy Dixon Salazar, and I want to highlight this one line here because I feel it's very powerful. Tracy says, "I thought the data would be shared. I thought it would be used to understand patient journeys. How does a child go from being healthy to progressing in this disease state?" But none of that happened. And that did not happen because we did not unify data at the patient level. We didn't have a shared identifier to be able to do that. Now, why would you need to link data at the patient level? Because of several reasons, and a few examples that I can cite here are you want to avoid duplicates in your natural history data. You want to be able to tie clinical trial results with the natural history data at the patient level. Or like me, you want to count the number of patients true number of patients that you have in your disease and not a hand-wavy guesstimate that no one could use. Or you want to connect your patient-reported data with clinical data from your EMRs, from your hospital systems, and get a holistic view of the disease. And there are so many other benefits. And I kind of focus at registries and natural history in this case, but it doesn't have to be. You can connect biorepositories to this. You can connect diagnosis companies to this. Anywhere, any part of the ecosystem that a patient interacts with generates data, and that data stitched together offers a more holistic view of the rare disease knowledge base than without. Let's dive into an example and talk about what shared identifiers mean, because I think this is the concept that a lot of people don't quite understand, and a lack of that understanding makes it harder for us all to have a shared conversation about data sharing. Let's say there's a John Doe in the Sanford Cards registry. There's information about this person in the registry, and I call this radioactive. I call this radioactive waste. You don't want to touch PII at all, personally identifiable information. That would be name, date of birth, gender, anything that can really uniquely identify a patient. And in the rare disease space, where you only have, say, 50 patients in a disease, you can very easily identify with just the first name, right? If someone says, oh, I know patient Raghav with the disease SSMD, that's my son. You don't even need anything else, right? And that's how sensitive this information is. Typically, patient registries do have this information because you want to be able to contact the patient back, and that's the whole point of it. Let's say there is a clinical trial running for this disease, then they would also collect some form of patient information, because somewhere in their clinical trial database, they need to know who this person is. And that's radioactive too, right? Some databases are uniquely equipped to handle radioactive information. Now, if you want to put the data together, if you want to connect your registry and trial data, you would share that radioactive information. You would really share identifying information between these two entities. It is possible to share identifying information under HIPAA if both of these institutions are covered entities and you sign a very large agreement called the BAA, which puts you under a lot of pressure and liability. But for the most part, most privacy laws don't allow you to share identifying information. And you don't want to do that because it is very, very risky and radioactive. Instead, you would want to share identifiers. Let's say there is a shared identifier, a random number that represents this patient on the cards registry, and the same identifier, the same random number representing the patient in the clinical trial registry. Then you can connect them together by sharing the identifiers. The identifiers themselves have less of a privacy burden when it comes to sharing, and there are more techniques to make them even better and safer and more privacy friendly. Now, let's talk about what shared identifiers are. The software developer in me defines an identifier as random characters. That is all they are. They don't have any meaning. All of these are identifiers, right? They're just random characters that I happen to type on this form. But identifiers get value when you add context to it. And in this case, when we talk about shared identifiers, these are identifiers shared between databases based on some rule. In the example that I showed you, we had a rule that said the same patient gets the same ID. As long as this rule is met, these two databases have a shared identifier and they can talk to each other whenever they choose to. To clarify the terminology in the rare disease context, there are many terminologies that we commonly use: GUIDs, EUPID, CRIDs, etc. All of these are shared identifiers that are built based on a rule that these identifiers are associated with a patient and not purely random. And that is all it is. So when when you hear the term GUID, although it's colloquially used in the rare disease space to refer to an identifier that represents patients, the technical term for GUID is globally unique identifiers. It has no meaning whatsoever. It's purely random. In the rare disease context, we use it colloquially to refer to an ID that is associated with a patient. Let's talk about implementing shared IDs in GUIDs. I'm going to use the word GUID and shared ID interchangeably, but I'm shifting my weight towards shared identifiers because that is truly what you want to get towards. And GUID is just one type of implementation of shared IDs. How do we generate a shared identifier? Going back to our example of having a registry, a patient in a registry and a patient in a clinical trial, we have a system. I'm going to call this an ID generation system. This is a piece of software. You pass the patient information to this piece of software, and that stores the patient information, maps it, it generates and maps a random ID to it, and that's the rule. The same patient gets the same ID, and that rule is enforced by the system, and it returns the ID back to the registry, and the registry stores the ID in the database. The same is the case with the clinical trials database. They send the patient information, they get an ID back, the same ID, same person, same ID rule is implemented here, and they store the ID in the database. Now, this is awesome, right? Now you start to have a shared identifier that's being built. The problem is patient identifying information has been exchanged here, and this is a big, big no in the privacy world. Now, there are a lot of solutions that are out there that implement privacy-preserving ID generation systems. You might have heard of the NIH GUID, there's the EU PID, there's a new Neuro GUID, there are commercial systems like Datavant, HealthVerity, CureSDev is building one, there's a CRID, there's one that's Pulse Infraframe is building. There are a whole bunch of these technologies that are all based on the ability to exchange a hash instead of a patient identifying information. So what they do is they take the PII, first name, last name, date of birth, city of birth, whatever it is, convert it into a random value based on an algorithm. More specifically, this algorithm is run by the data owners. So let's say you have your database in the RAREX database. They would take the patient identifying information, they would use the hashing software provided by the ID generation service, convert the PII into a hashed value. Some people call it a token. It's the same thing. You send the token to the ID generation service, and that creates a unique ID for you based on that token. And that ID is stored back into your data owner system. So going back to our example, instead of exchanging John Doe's personally identifiable information, CARD's registry would exchange a hash, a token of it. The token is what gets stored in the ID generation system, right? And it still generates an identifier for the patient and returns it back, which gets stored in the CARD registry, similarly with the trial database. As long as both these systems use the same hashing tokenization software, they should be able to get the same identifier back. So as long as CARD's registry and BridgeBio, in this case, in this example, use the same software to generate these IDs, the software enforces the same patient, same ID rule, and you get the same ID back. And now these two data sets are now linkable. They have the ability to be connected at some point down in the future. The International Rare Disease Consortium recommended in 2017, believe it or not, recommended privacy-preserving record linkage algorithms as the best algorithms for data exchange in rare disease space. The class of algorithms that I'm talking about that use hashing are all privacy-preserving record linkage techniques. I'm going to go through a few of the implementations without getting too much into the details, because a lot of them have the same conceptual structure that I explained in a second ago. The NIH GUID was one of the first people to come up with this technique, and they use a hashing technique that's different from some of the other folks. Their implementation is a little bit more privacy sensitive in the sense that your inputs have to be exactly right. So if someone misspells my name with 2 S's instead of 1 S for my first name, you're going to get a completely different ID. But that's okay for the NIH GUID system because that's the kind of tolerance they have. But it might not be okay when you start scaling to a larger patient population. There's a NeuroGUID, which has taken the NIH GUID technology and adopted to the Neurobank ecosystem. There's the EU PID. The EU folks developed an algorithm that's very similar but different. This offers the ability to re-identify a patient if there is a trusted third party. This also offers the ability to pseudonymize their identifiers. So the chance of re-identification goes down. There are a lot of cool techniques that they had implemented, but their hashing algorithm is still not as tolerant to misspellings or missing fields and so on. There's a commercial company called Datavant. They have a token generation process. It's the same thing. They have a hashing structure. They have a hashing algorithm that's a little bit more tolerant to misspellings and can take arbitrary data. They do some encryption on top of it to generate the site-specific tokens. There's another company called HealthVerity, which leverages Bloom filters. This is an implementation detail, but Bloom filters give you more tolerance to misspellings and other things based on the way they're implemented. My company, CuresDev, essentially generates patient IDs to make the datasets linkable using Bloom filters. We also have a lot of privacy guarantees based on differential privacy, which is a provable privacy control mechanism. I'm also building all of this as fully open source and an open standard. So it is not just me that benefits from it, it is the entire community. A part of being open source is making sure the community can continue operating on these IDs, even if CuresDev, even if I go out of business, even if I don't have the mental bandwidth to work on this company anymore. That is why we wanted to make this public and open source. I wanted to just talk about some caveats of some various caveats of different systems, right? Each system might exhibit one or more of these caveats, but I wanted to quickly go through them and for you to have a nuanced understanding of these systems. Some of these systems work only in the US. They might not tolerate as much misspellings. If the databases don't use the same ID generation system, they will not get the same IDs back, which defeats the whole point of it. Re-identification becomes a challenge if you store the actual master patient identifier in all of these databases, because the moment you combine the databases inadvertently, you inflate the information available and make re-identification possible. Some systems actually store patient information, even though it's encrypted. It is not safe because, as we all know, the decryption key lives very close to the patient information, and so an attacker can decrypt it. I went through the comparisons of all of these systems already, so I wouldn't bore you with the details. But once you get the slides, you have this to review. And with that, I'll stop.
Awesome. Thank you so much, Sanath, for that informative presentation. We're now going to move into our panel discussion and Q&A. So with us today, in addition to Sanath, we have Dr. Daniel Lavery. He's the Chief Science Officer from the Lulu Foundation. We also have Michael Barrow. He's the Chief Technical Officer from Pulse InfoFrame. And finally, our very own Alexandra Batourné. He's the Executive Director with Critical Path Institute.
Thanks, Dominique. And as a reminder for everyone listening, please type in your question in the Q&A. We're just going to do some comments and then address your questions live. So thanks so much. And that was very compelling and also moving. You know, as RDCA-DAP, we try to build this aggregation of data and try to provide that place for scientists all over the world to be able to leverage as much as possible out of the data we're able to get people to share with us. And so I talk a lot with foundations and rare disease community. And there are a few things that I always say the first time that I meet with the foundation or registries that is starting is, you know, or new communities, you know, like first thing, like you need to have that engagement with your community. You know, you need to get as many people as possible at the table with you to talk about sharing your data somewhere and try to build disaggregation of data for the sake of leveraging as much information as possible. And then there are a few other things, and one of them is you need to think about prospectively linking your data, and you need to think about this de-identification and patient, GUID, as we say improperly, apparently, questions. So I think that was very relevant, and I, often get a lot of questions. So first, I would just like to get Dan to introduce himself and the Lulu Foundation further, and then please talk about, why did you make, this early choice of adopting a GUID for the Lulu Foundation? What were the reasons behind that? Why do you think it's relevant? And also talk maybe some of the issues that you faced when you were working toward that selection.
Sure, great. Thanks very much for the invitation. Happy to be participating with this wonderful panel. And thanks, Sanat, for the very clear and very eloquent presentation you made. And it's very touching to hear your own personal story. I'm the Chief Scientific Officer at the Lulu Foundation. The Lulu Foundation was founded by a family who themselves was affected by a rare disorder, CDKL5 deficiency disorder, which similar to the disorder that Sanat described, monogenic disorder that leads to neurodevelopmental delay and largely intractable seizures. One of the biggest challenges we have, and this goes to the parable that Sanat mentioned about the visually impaired people and the elephant, but for the CDKL5 community, imagine that elephant is spread out around the entire world. Wherever you're going to find kids, you're going to find kids with CDKL5 deficiency disorder. It's largely generated through de novo mutations, and therefore even more of a challenge in trying to identify all of our patients and getting them all organized. The biggest challenge as a community was the fact that we had multiple different parts of the elephant in Australia as a database data collection effort spearheaded by Dr. Helen Leonard. There was an NIH-funded natural history study for some time in the US. There is now a clinical registry for CDKL5 patients in Italy, and we have some ongoing biosample collection efforts in Ireland and Italy. And the point is, how can we bring these all together in a way that allows us to be able to leverage our community's organization and not be suffering from this silo effect, which is in a way that the silo is almost the default that we see for all of these different data collection efforts, which is why you need to work hard to align all the groups in order to get everything in place as soon as possible in order to allow the alignment and merging that Sanat referenced. It's not an easy thing to do. When we first started off, our advice from the US clinicians was just use the NIH system, their GUIDs. The NDAR GUID was one that was suggested at first, but we quickly realized that this was going to run into severe problems with the data collection outside of the US. The ability to ping servers was strictly restricted, and therefore we really needed to have another solution. In fact, that was such an issue. I know another rare disease community had even decided to forego collecting data outside of the US because of all the challenges they had with being able to use, colloquially, the GUIDs to be able to align data. And that's something that we cannot do with our international patient population. We're not as ultra rare as the disorder that Sanat's son is living with, but we're what we would consider medium rare, in that we probably have tens of thousands worldwide, thousands in the US, but only a few hundred in the US who've been identified. So we need to be able to leverage as much as we can of all these different studies in order to bring them together. And there are the usual challenges one can imagine. Sometimes, there may be academics with concerns about, being able to publish, et cetera, but those are easier to deal with once we actually get our community engaged. And so when we set out to establish the CDKL5 registry, the first thinking that we had was we needed to have a system that would allow for alignment and merging of data internationally in a way that was going to be fully compliant with new and emerging regulations, and also in ways respecting the rights of the families to control where their data are shared. And so that's what really led us to the collaboration with Pulse InfoFrame to provide a truly international solution to this problem of de-siloing this critical information that we really need to assemble in order to help our families.
Thanks, Nad, and nice lead into Mike. So Mike, I'll let you introduce yourself and Pulse Info Frame, and also, talk about your perspective as, a bigger collection effort, commercial entity. What are the particular considerations, criteria, issues that you are dealing with as it pertains to selecting a de-identification system?
Sure, thank you. I'm Mike Barrow from Pulse Info Frame. Pulse manages several rare disease registries, including one from CDKL5 for the Lulu Foundation, and that's how we got involved in this particular topic. I'll talk just a little bit about some of the technical details, because that's my perspective on this whole thing, and about identifier storage and generation. And from our perspective, we're really concerned about the mechanisms by which we can do this for real today, solving very particular practical problems with particular institutions and people who have data that we want to contribute. And how do we do this in a way that doesn't get us into trouble with regulators? In particular, because we in particular are exposed to things like fines from European agencies in the United States in case if we violate privacy regulations. So we have a very practical concern about this in terms of, well, what can we do today that actually works, even if it doesn't solve the long-term constraints that Saneth very clearly described. So what we've done is built a system fairly similar to an NIH system, but we expect it to be used by people we know, and that we talk to in advance, such that we can structure the process of creation of GUIDs or identifiers in a way in which we get a consistent results. And basically, as Saneth said, we essentially use a hash, even though we call it a GUID. The hash is subject to mismatches if the data is not consistently entered, which isn't the worst thing in the world, but you want to minimize mismatches. So one of the things you want to be able to do is to avoid duplication. This is one of the consequences of a mismatch is you can have data from the same patient and not realize it. But by the fact that we manage the small set of people that are actually entering data for us, we can control the mismatch problem. And we understand that that's not a long-term permanent solution along the scales that Saneth was presenting. So I'm glad that he's explaining that to the rest of you. In terms of the actual mechanics of generation, there's an interesting problem, which is in many places, it is not legally possible for the personal information needed to generate the hash value to leave the country. And so that means you can't build a server in the cloud in the United States, or in our case, Canada, that works worldwide and is compliant. If the computation, even if it's a transient computation, occurs on a machine that's not in the country of origin. So one of the solutions we have this is even though our solution, our computation is accessible through a web browser, the computation actually runs in the web browsers and it never gets transmitted to our servers, even though we host the code. The problem with that is the complexity of our analysis and creation of the GUID is limited, because we can't run a whole long, huge, complex piece of code in your browser to make that happen. And so it's one of the things we would certainly be looking for in the future to be able to take advantage of to make a more generalized solution. Other thing I just wanted to reemphasize what Saneth said is the idea of linking in anticipation of emerging later is a very important concept. It doesn't mean that if you don't link today, your data can never be linked, but you do need to link as soon as it's reasonably possible. And if you need to link differently because we have a better standard in the future, you can do that too. But it is the issue of generating the identifier and associating it with your data such that you can then, when opportunity for sharing comes out, you don't have to solve this problem at that point. That makes all the difference. I know from academically, people trying to share data, they really do spend months and months and months talking about, I've got 10 records and 20 records, why can't we just throw these together? And for all the reasons we talked about, there are just legal constraints and legal liabilities, in addition to just wanting to follow privacy laws, because we ought to, that just get in the way. And so like trying to solve these things early in the same way that everybody else does means it doesn't have to be a 6-month project every time for the rest of humanity's existence. So,
yeah, thanks, Mike. Those were all great points, and to your point about linkage, and really Sanath's presentation insisted on that. It's, people, you tend to think, okay, GUID, privacy, it's also about linkage and not losing data. And for us at RDCA-DAP, we always get the question, are you concerned about duplication of data? Because we deal with multiple contributors with not necessarily a centralized conversation. Duplicate is usually not an issue, and we have means to get around that when we do the analytical work that we do at C-Path. My main point is, well, the real problem, we may lose longitudinality of data, and we all know how important it is to be able to track patient progression, and losing that data is really an issue. Sanath, I want to take it back to you before we start on the Q&A. We have tons of questions coming in. So we're collaborating with Pulse InfoFrame on some project that deal with international aggregation of data. And so it's just leads me to some of these points on the global piece. Is there a path to a global solution? My sense is, you're going to tell me we're going to work toward that. So the second question is for all the foundations and registries that have not adopted the system yet. They're facing that diversity of potential choice. So where do they take it from there? Like, we've seen a few solutions, there are a few options. You're more research center based. You may want to select the neurobank, neurostamp approach that deals with scientist information exchange. You are an ultra rare disease, your disease with more patients, like where do we go next? So 2 questions in one. Is there a global solution that we can work on so that it's simpler? And in the meantime, what do we do? How do we inform our selection as a foundational registry that hasn't adopted the system?
Yeah, I'll answer the short and the long of it. So right now they don't exist, but they will, and they can. Both global and also your ability to not get logged in to the choice of GUIDs or identifier technology you choose today. And here's why. If at all we learn one thing from history of humanity, it is that history repeats itself. People will take a system that someone built, rebuild it, make it better. And you're always going to be left with a plethora of options. And optionality is great for the marketplace, right? It's great for people, great for patients, fosters competition, and so on. And the moment we get logged into one, it actually closes down optionality for people. So for me, if I were to just sort of explain a solution here, I think it is very similar to what Mike said about running the computation of hashing and tokenization at the edge, at the browser, at the client side. But rather than doing it on the browser, we would do it on a server located in the country. Like AWS, for example, offers 31 data centers, right? I'm assuming they will continue to offer more data centers in places where they have different privacy laws, because they would have healthcare customers, financial customers that have to comply with unique privacy laws. Cloudflare offers like edge servers, right? You can run hundreds of edge servers across the world, and you can run computation on them. And so, we don't really have to take the data out of the country, out of the locality into a remote server. We could actually bring the computation close to it. And that's how you go global. The second question around how do we not lock in to a particular choice is interesting. And this is where the open source comes in, right? So I don't expect everyone to get standardized on a technology, even if it's open source, right? I expect the open source technology to have the ability to chain a lot of different identifiers together. So let's say you start with the NIH GUID today, but you have enough information to add another GUID on top of it, right? And you do that in the future because that system offers some more, superior guarantees and privacy protections. And in the future, let's say a particular country's government mandates everyone to using their GUID system. You should be able to add on to it, and we will be able to create technologically solutions that allow you to not have the lock-in problem, because that is what we need as a community to evolve. And once we get that technology in place, and once the technology is open and shared with a lot of people, you would see 20 different GUID systems pop up with a whole different set of capabilities and features that just gives optionality to people. And we will eventually standardize on a few big ones, but there will be choices which people don't have today.
Thanks, enough. Mike, Dan, unless you have a comment you would like to make, we can go to the Q&A.
Let's go to Q&A.
Yep.
All right.
Exactly.
So tons of questions. I think there's a couple of questions that are related to how do you handle data that have been linked using different systems. Two databases are using two different softwares to generate the idea. Will they be able to communicate or not? If you want to link registry with several databases, is it possible to use a different generation system to generate several identifiers for each patient? Globally, that's a couple of questions around that.
I mean, I'd like to get Mike's take on it as well, but my take is yes, as long as you have enough patient identifying information that you can link to. So let's say you have a registry that has a GUID, NIH GUID, but then you also have a clinical database at your hospital system that also uses the same NIH GUID. Now, your registry might not have enough information. You might not even store patient information, but you can hop into the clinical database to grab the patient information to regenerate a new ID with a new generation system, right? And I think the whole point of these IDs are linkability. So at some point in the linking chain, you will end up with a system that has patient information so you can go back and generate a new ID.
Yeah, the sort of the practical answer to that is you can always, if you can get enough information, you can always build ways to establish a link chain. I won't lie and say it's necessarily easy, and the most probable impediment is access to technical people who can actually set up the system and maintain it for you. It's the advantage of having a single system where for the generation of the IDs is that you don't have to have the same storage systems. You can have different companies all providing completely different style systems for managing data. If they can at least agree on an identifier on an edge level, then it makes it easy to link things together. And even if they don't, as you say, you can do it mechanically, but the people are involved are left to do it themselves, which is technically doable, but probably too expensive.
And I would agree, just to chime in with a real world example. The Pulse team and I were working with Dr. Helen Leonard to be sure that we had both collected the same sorts of information that would be required to generate the GUID to allow the data to be, in this case, aligned more than merged, and to be able to share the data at that proper level. And it was a quick discussion. It was not esoteric. It really were basic demographic elements that were found in both. But we did have to confirm that, for instance, the parsing of the data, whether it's middle names or initials, et cetera, would be consistent between the two in order to avoid some of the issues that were mentioned before. But it just takes that sort of confirmation to be sure that those same data elements are present in the same format in both collections.
Thanks. I have another question pertaining to IRB approval. It is my understanding that each study trial will need IRBs to approve the use of GUIDs and collection enough to generate a GUID for the patients, and for the patients to consent to the sharing of GUIDs. Was that clear, or maybe I'll just read the question again?
Yeah, I think you need IRB approval.
Okay, yeah.
Because you run the risk of re-identification. Let's say you have, and this is a scenario that I'm always worried about is you have a hospital system and you have a registry database, right? Both of them have GUIDs right now. The hospital shares with company A and the registry shares with company B. For some reason, company A and company B decide to share with each other, right? Now, at that point, unbeknownst to the original collectors of the data, there is more information that can potentially re-identify the patient. So the moment you have shared identifiers that are truly shared, you run the risk of re-identification unless you have some way of controlling how the downstream sharing actually happens.
Thanks. I think now we're going to go more into the nitty-gritty of the questions. So I have a question from Jessica discussing, I think you touched about that a little. They face issues with the consistency on how the names are entered. For example, Anna Kate Smith, and then Anna Kate, middle name blank, last name Smith. Now these two entries generate two identifiers. And she would like to know if there are any suggestions you can make on a generator that's flexible enough for this.
Yeah, I mean, there are different generators that have different, flexibility levels. But I think, as Dan said, it's better to align on the data entry, so you don't have to have these challenges. If you're collecting data from a real world system that you don't have any control over, then you need a system that's more flexible.
Okay. Another question around what are the suggestions you guys may make to be mindful around API connectivity and developing the workflow to start the identification implementation? Maybe, Mike, you have some input on that?
Who asked that question? I just want to read the text.
It is a Brittany in the Q&A.
Okay. Yeah, I guess so. I can imagine all sorts of different problems. The main issues around APIs that I can think of are not really technical issues; they're regulatory issues, as I mentioned before. And you just need to make sure you know where your data is flowing, even if it's not being stored. In terms of the transmission of the data, I mean in any of these systems, these are largely just binary, blocks of data, and transferring. Shouldn't presume any particular API issue. I guess one of the sort of technical issues that we ran into is because of the word GUID, many people might assume that it has a particular size, either 128 bits binary or 36 characters typed out with certain formatting, where it's just an arbitrarily length size token, which could be a little bigger than that, or a lot bigger than that. And so just sort of allocating enough space for the identifier and checking in advance to make sure you know what the limits are, some of the practical matters that come to mind. I'm not sure I really addressed your question, but feel free to narrow down if you.
It seems to me, well, we can always send you follow-up questions. I have a more general question from Jim from the Steve Syndrome Foundation. Jim is asking, what about biorepository? Is that changing anything to what you presented, Sanaf? And maybe more globally, are there any specific considerations that are dependent on the data type?
So technically, nothing should change. As long as you have enough information for the patient, you should be able to generate an ID. But I guess Dan and Mike maybe can, if you have experience, can talk about other aspects of it.
Totally agree with Sanat. It's absolutely essential, in fact, that we're able to connect these different sorts of data, because for us, the power of the biorepositories, whether they be serum samples, CSF samples, cell lines, with clinical data, is absolutely essential to our ability to interpret any of the results. And so we're very, it's a real drive for us to be able to share the GUID generating system that we're using with Pulse, with any of these partners and able to allow that. So it doesn't matter for us whether it's a blood sample or it's a a clinical file, an EEG. We want to be able to be sure that we have that unique identifier to be able to link that set of data with the other data sets around the world.
Yeah, it sounds very relevant, and I had like recent conversation of potentially like contributor with multiple data types, and RDCA-DAP is not a biorepository. So, this is part of the conversation right now. Like we need to work ahead of, like the sharing conversation is great and there's a path, but we need to do some groundwork ahead of sharing so that we don't lose any information. I have another question from Nisi. Currently, they found an 8% error rate when they generate IDs. Is anyone using fingerprint or blood or facial recognition or any other options to improve the error rates when creating a hash?
Well, the error rate you're talking about is probably coming from the GUID system itself, like the GUID implementation itself. You don't need additional information, especially we have commercial systems that actually work at the US healthcare population level, like 30 million patients, 300 million patients, right? They don't have fingerprinting information to uniquely identify patients. All you need is enough bits of information and a good enough software that can tolerate these errors.
Thanks, Anaf. We're getting to the top of the hour, but I do see some questions about re-identification, and we talked about that when we prepared the call. Can we touch about re-identification issues? Is it a real solution that is going to prevent re-identification? If you guys want to comment on that before we wrap up.
I mean, I personally feel like it's a real issue. It's just to give you an example. I used to work at Amazon, where we were building a very similar system internally, and we were trying to make sure two tables don't get connected with each other, because that's how paranoid we were. And when you talk about two tables owned by the same company, it's one thing, but two pieces of information owned by two different entities that were collected at, potentially the decades apart, right? That have complied with different privacy laws. You're just increasing the problem to a great level. So as long as you have the same identifier shared across these systems, you are going to run the risk of re-identification, unless you put some controls in place to control when those data gets merged.
Yeah. Thanks. Any other comment, Mike or Dan?
Yeah, I would just a quick comment. It's yes, re-identification is a real problem. It's a real legal problem for compliance, because especially GDPR, you cannot truly ever de-identify data. I mean, it is not humanly possible. If you, the only thing is you can do is change the probabilities of re-identification. And it's a problem that's not going to go away. And as we start sharing more data, it will be a more and more pressing issue. And there will never be a single technical solution, by the way. But there will only be ways to make it smaller and smaller.
I would say, just adding in, particularly in the rare disease community, this is even more of an issue. Sanat's example of his son, that, if you're identifying the mutation in the GPX4, of a patient in the US, it was going to be right of. So just we are very sensitive to that, and that's why we do have multiple levels of regulation, both within our IRBs and through compliance with GDPR and other regulations, that we truly need to do this to protect our patients.
Great. Well, thank you very much all for joining and helping us with this webinar today. That was great, and we had tons of questions we couldn't go through, so we'll try to compile them and see if we can contact you to address the pending questions. In the meantime, you can always contact us. You have the emails here. Thanks again, Tanaf, for the presentation. And we will see you in June for another webinar under the RDCA-DAP webinars this year. The next one will be on patient consent. Thank you all for attending.